Hello, I'm Laura Cassidy from the American Chemical Society. Welcome to this media briefing from ACS Fall 2021. We're joined today by Dr. Stephen Townsend and Rebecca Moore from Vanderbilt University. They're studying sugars from human milk in plants that could help treat and prevent infections in newborns. Dr. Townsend?
Yes, thank you for the introduction. I think the best way for us to start is for me to briefly tell everyone a little bit about our team and our science. From the scientific perspective, we're a chemical biology group with a focus on carbohydrate science. Now, if you, were to ask us, what do we care about and what do we do on a day-to-day basis? I would tell you that we care about why people get sick, particularly women and children. And ultimately, what we try to do is use some combination of chemistry and biology to address problems that are related to those two areas, both fetal, infant, and then ultimately maternal health. Now, although Rebecca will give you, details about the specific project we're going to talk about today in a moment, I'll tell you that we've been on a 7 to 8 year voyage in human milk science, specifically trying to discover how a woman is able to protect her baby from getting sick. Now, what I think is special about our approach is that we don't really stop there. What we try to do is take that knowledge and then ultimately use human milk and its components to solve problems in health that are completely unrelated to breastfeeding. So that's what the project that we'll be discussing today.
Great. So I feel very fortunate that I did come into this program as it had already been started, in which predecessors in the lab had looked at how HMOs, or these milk sugars, really reacted to cells in vitro. And so what we initially found in our lab is that this mixture of human milk oligosaccharides is really able to inhibit both growth and biofilm in GBS infections in cells. So when I came in, I was really interested in looking at more of the immunological response to how HMOs react to GBS infections to cells, both ex vivo, and then also looking at an in vivo mouse model. So where I came in is knowing that these milk sugars had these antimicrobial properties. I wanted to see if we could then prevent infection on maternally derived tissues, as well as these immune cells that are found only in pregnant women. And really excitingly, what we found when we were looking in cells, they were able to prevent this growth in biofilm, is now that we're looking at tissues, we were also able to prevent this adherence to these gestational tissues derived from pregnant mothers. So knowing this, then we wanted to take these results and then look at them in an in vivo mouse model to see if we could then prevent infection, ascending infection through the reproductive tract with HMOs against GBS infection. And just as excitingly as before, we did find that across 5 tissues in the reproductive tract, we saw this significant decrease in bacterial burden across all these tissues. So really moving on from here, we're really just trying to find out more what's going on with the immune response, and that's what's coming up next now that we've gotten more information looking at tissues derived from mothers and in an in vivo model, not just looking at cells in culture.
Thank you, Dr. Townsend and Rebecca. And now I'd like to ask you a few questions about your work. First of all, what gave you the idea to investigate these human milk oligosaccharides, or HMOs, for antibacterial properties?
Yeah, I guess I can start with this one. So, this was sort of my baby when I first started as an assistant professor. And really the idea came from just looking at the macromolecular differences between human milk and infant formula. So if you really put, bovine milk, which is the basis of formula, head to head with human milk, and you compare the different ingredients, what we see is that there are a great deal of complex oligosaccharides that are present in human milk, and they're absent from bovine milk. So once we started to follow that initial trail, we realized there was a lot of special properties that are affiliated with human milk that you don't see in bovine milk. And the hypothesis was that many of these were attributable to human milk oligosaccharides.
Okay. What types of problems does Group B Streptococcus, or GBS, cause in infants and adults?
So I can take this one. So in a healthy individual, so this is going to be the majority of your population, it really manifests asymptomatically. So most people wouldn't even know they have GBS, but where it comes into play is that up to 50% of pregnant women will carry GBS at some point, with around 20% of women testing positive at the time of delivery. And so it's not going to affect the mother, because she's typically asymptomatic, but she can transmit it to the baby through vertical transmission during labor and delivery. And then this is a major cause of neonatal sepsis, meningitis, and even infant mortality. So really, it's more for those with the weakened immune system and not your healthy individual.
I see. How are Group B Streptococcus infections currently treated or prevented in newborns, and what are kind of the drawbacks of the current treatment?
Yeah, so the CDC currently has a roadmap for treatment that involves intrapartum antibiotic prophylaxis. Effectively, what that means is that during week 35 of pregnancy, we know that most women in the United States will be evaluated for Group B strep colonization. About half of women in total will have Group B strep over the course of their pregnancy. At the time of labor and delivery, about 20 to 30%. So the reason that we have sort of some control over this problem in the United States and a number of Western countries is that if you are Group B strep positive, you have an antibiotic for the entirety of labor and delivery. But we know that that can be problematic, right? Antibiotic prophylaxis and use in general is not without its problems. So there's always going to be the fear that we start to encourage Group B strep to evolve resistance to the antibiotics that we have that can treat it. We've already seen it. When I was born back in 1983, my mom was given erythromycin to prevent Group B strep transmission from her to me. In 2021, we no longer use erythromycin, right? Because Group B strep has evolved resistance so quickly. So I think that's the big problem that we face when it comes to treating this pathogen. We only have 26 or 27 antibiotics that we use in the clinic. Group B strep is already not susceptible to many of them. It already has resistance inherently to many of those antibiotics. What happens when we run out? I think that's sort of where we're looking at when we try to develop new solutions.
Right. And do you think bacteria would be less likely to evolve resistance to HMOs than antibiotics?
I would say absolutely. A common path for treating infections now, just all bacterial infections, is looking at the use of an adjuvant. So that means a component in addition to an antibiotic, so that the bacteria has less chance of evolving resistance if it has two drugs going at it rather than one. So that's been our plan, or one of our plans in the beginning was looking at HMOs to help potentiate some of these drugs that have already gained resistance.
I see. Okay. And are you also looking at using HMOs just by themselves without other antibiotics?
That is the ultimate goal.
Okay. How many different HMOs have you tested in your two species microbiome system so far?
So in the two species microbiome, we've looked at 6 different individual HMOs. We've also looked at the HMO cocktail, which is typically how we've been looking at HMOs in general, not as an individual. They work better when they're with other HMOs because that is how they're present in breast milk. So we've been looking at the cocktail a lot, as well as some plant oligosaccharides. So one additional plant oligosaccharide as well.
I see. Okay. And I understand that HMOs can have different effects on good bacteria versus bad or harmful bacteria. Can you explain that a little bit?
Yeah, so I think this is sort of an evolutionary wonder. If we think about back in the 80s and early 90s when people first started studying HMOs, they realized that they were prebiotics, right? They can go in and selectively grow good bacteria over bad. This is because good bacteria possess the enzymatic machinery to metabolize HMOs. They're just a source of carbon, whereas there are very few pathogens that can do the same. So basically, mom provides a selective growth advantage for good bacteria over bad. Now, this is actually, why our group has sort of been able to make a living, because we figured out that human milk oligosaccharides are antimicrobial agents, right? So if you flip that coin over, on one side you have bacteria that can selectively grow that are good. The other side, these same compounds are able to place evolutionary pressures on bad bacteria, right? So it's pretty cool. They both promote the health and growth of good bacteria and can eradicate bad bacteria.
Okay. How does the antimicrobial activity of HMOs compare to, conventional antibiotics? Are they as strong or not as strong, or how do they compare?
Well, specifically just looking at Group B strep infections, when we're looking at physiological concentrations. So the concentration is higher than your typical antibiotic, but it is what is found in human breast milk. And we have found that they are as potent as an antimicrobial alone. But additionally, they're able to increase the efficacy of just our known antibiotics, especially the ones that have resistance mechanisms in place.
I see. Okay. Can HMOs be made synthetically, or do they need to be isolated from breast milk?
Yeah, so our group and a number of others in the organic chemistry community have synthesized human milk oligosaccharides. Now, if you ask, is that the way that we're going to, get these compounds out and about to the public? I would say that the answer is definitely not. So when it comes to synthetic organic chemistry, what we can do really well is make complex structures, and we can make tool compounds. This is something that's still prohibitive in other areas of manufacturing. But if you want to say, how do we get, swimming pool scale quantities of the most common human milk oligosaccharides? Well, that's going to be in the microbial host. So there are a number of companies that are able to manipulate yeast and E. coli in order to produce these compounds. So that way we can get hundreds of thousands of kilogram scale available.
Okay. Do you think HMOs could someday be given to adults as well as babies in place of antibiotics?
So this may be a double answer from both of us. I'll tell you, I went to consult at a company, and I won't say the name of the company, but there were several people in the room who were over 50, and they all had intestinal types of issues, whether it was Crohn's disease or some other type of inflammation. And during tea time, I noticed that they were adding something to their tea that wasn't sucrose. And it turns out that they were eating human milk oligosaccharides. Right. And so this was a non-scientific study where they were hypothesizing that these compounds they were making could very easily help treat their Crohn's disease and other types of intestinal inflammation. My suggestion was, hey, you can, fund us and we can put some science behind that. So I think in the future, the answer to your question is yes. And that's where we're going. We're trying to figure out what uses for these compounds exist that we're not even thinking of, that are outside of the sort of sphere of infant health and wellness.
Sure. What are your next steps for this research?
So twofold. When looking at the two species microbiome, we definitely would like to look at other pathogens as well as other commensal species. So far we've only looked at two species, one pathogen, one commensal species. Obviously the microbiome is very complex and we see multiple species. So we would like to look at two additional species as well as other milk sugars, other plant oligosaccharides, and their interactions in this microbiome. So that would be one half of the project. This is just in the infant stages of looking at the microbiome with the HMOs. And then the other half of the project is really digging deeper into the mouse model and how HMOs could be used to prevent infection, preterm birth. Can these be used to treat infant infections or, as you're saying, mother infections? So it's continuing now that we've looked so much in cells. We're now more interested in looking in applications, so into our in vivo models.
Very good. All right. To conclude, what do you want viewers to take away from your talk? What's the key conclusion or message?
Yeah, we're a group that's focused on human milk science, right? And we're trying to really unearth how it is that mom is able to protect her baby from the level of chemistry. It's a very mechanistic question. We also want to, bring these compounds to the attention of the public so that we can find just awesome uses for them, right? There are disease states that we can probably treat with these compounds that are non-toxic. Babies eat somewhere between 8 and 20 grams per day. So I think they hold a great potential. Along with that, though, I would say that everyone should keep in mind that breastfeeding is the gold standard. It is incredibly important. And by no means are we trying to shame people who formula feed, because people have choices they have to make in circumstances that we don't know. But we're also not trying to replace breastfeeding. So at the end of the day, what we're trying to do is provide science and tools to the community who fund our research, but we're not trying to replace breastfeeding.
Yes, exactly. I think we know that breastfeeding will always be the gold standard for infant feeding, but for those who can't, we want to better understand the processes that are going on so that we could create better formulas and just better understand the processes for those that can't breastfeed.
Thank you, Dr. Townsend and Rebecca. Media briefings for ACS Fall 2021 will be posted throughout the meeting at www.acs.org/ACS Fall 2021 briefings.